Thank you very much for giving me this opportunity to talk about works. Today, I will talk about one of the synthetic biology area by incorporating a natural component into DNA to expand the genetic alphabet. And also we talk about the application, so what we should do using this technology. So there are a lot of creatures living on Earth. Actually, this is Roy Romelsberg. This is the next speaker, and this is my pictures. So although these are very similar, but all creatures are very different from each other.
About the same intellectual level.
I think so. So, but these have all DNAs with the same chemical components of 4-base nucleotide, AGCT, as a genetic alphabet. This is very amazing because only 4 letters are ruling our lives. And my question is, if we have more than 4 letters, so could we improve our functionality much more? So this is the simple question is also our objectives. So to do this, we have to develop a third base pair. We call it a natural base pair. To this end, this unnatural base pair can function as a third base pair in replication, transcription, and/or translation. And if we can make such a system, we can introduce new component into nucleic acid and proteins, resulting in increasing their functionalities. So, for example, this is a usual genetic code with 4 letters. But if you add 2 unnatural letters in this table, so you can increase the genetic code by 216. So you can add so maybe 100 amino acids into proteins. So as for nucleic acid has also versatile functionality. It's very amazing because they have only 4 very similar chemical compounds or bases. But the functionality might be less than proteins because protein has 20 standard amino acids. So this is nucleic acid with very 4 similar components. But if you add more different component into these tools, maybe you can make a much functional nucleic acid. So this is our purpose. So, but until recently, such an experiment is just science fiction. This is my favorite picture of US TV X-Files. So, but this is very interesting because the researcher tried to examine the alien's DNA. And this is, I don't know actually, this isn't these aliens, but she said there is a gap on the sequencing band. So that shows extra bases in the DNA. And this is just science fiction, but our sequencing method, involving a natural base pair, is imagined from this science fictions. Later I will show you. So, and at present there are 3 unnatural base pairs of 3 groups are functions in PCR amplification or transcription. And one is Floyd Romelsberg of Script. He is here, and he's talking about this So, and our group and Stephen Benner, there are three groups. So that I just, this time, I would like to focus our unnatural base pairs. So, more than 18 years, we are struggling to create unnatural base pairs by improving step by step. And I will talk about the latest, our unnatural base pair, DSPX, that exhibit high fidelity and efficiency in PCR amplification. So this is a DSPX chemical structures. And they have no hydrogen bonding interactions between pairing bases. And these shapes are different from those of natural bases. But we designed these natural base pairs by strictly refine the shape complementarity between pairing faces. So the shape of DS, so the DS base has a thiophene at this position. So the shape is very large from those of A or G. And in contrast, PX has a 5-membered ring of the basic part, 2-nitropyrrole. So, and T and C has a 6-membered ring. So that's why the PX is smaller than T or C. And in this case, so if DS paired with T or DS pairs C, they crushed each other sterically. So, and A and PX, in this case, just shape is slightly fit each other. So that's why we add the nitro group at this position. The oxygen, so electrostatically repelled this nitrogen of A, so that it can not pair each other. Instead of A, DS has no nitrogen at this position. So that's why the shape of DS completely fit with the shape of PX. So that's why this DSPX pair can function as third base pair. First, we try to know the ability of the DSPX pair. We performed 100 cycles of PCR using these unnatural base pairs. We first make like DNA containing DS by chemical synthesis and performs 100 cycles of PCR. But PCR is mostly after 20 cycles of PCR, the amplification is finished. So that to maintain the exponential amplification, we first carried out 10 cycles PCR, then diluted. Then again, we performed 10 cycles and dilution. We repeated 10 times these cycles to perform 100 cycles of PCR. So then we obtained a huge amount of DNA. It's so 10 to the 27th fold increased. It's okay.
Which polymerase was used?
Okay. In this case, we use a deep bent DNA polymerase, which has a 3' exon nuclease activity. Yeah. So it depends on the polymerases. Yeah, that's right. So next, so how much unnatural base pairs included in the final amplified product? Then we performed DNA sequencing. This is a Sanger sequencing, but we add the unnatural base substrate in the sequencing, but we didn't add the deoxy diterminator corresponding to the unnatural bases. So that's why the unnatural base position we can see as a gap like this. So after 100 cycles of PCR, the sequencing pattern is mostly same. So after 100 cycles of PCR, so the unnatural bases are still survived. And actually, this is a X-files sequencing like this. More detailed experiment, maybe Huai Romesberg will talk about quantification of to determine the selectivity and efficiency. So that I will just omit more detailed experiment, but we just tell about the result. Finally, we found the selectivity of the unnatural base pairing is more than 99.90%. This is using deep vein DNA polymerases. So, and another important thing is misincorporation of unnatural base substrate opposite natural bases in template. In this case, the misincorporation rate is less than 0.005% per replication per basis. So this is a natural base misincorporation rate. So that's why this unnatural base misincorporation is mostly same as natural base fidelity. So, and PX has a very unique because we can attach to any functional group the dispositions like diols, azide, and ethinyl. Azide and ethinyl can be used for quick reactions to modify to attach other functional groups. And also biotin can be attached to this position. So then, and when we use these DS and modified PX bases, the PCR amplification is very high. So we now have such a third base pair, so that we are trying to several applications. Then, and today I just will talk about the DNA aptamer applications here. So I just a bit talk about DNA aptamers. And nucleic acid aptamers single oligonucleotide fragments that specifically bind to target molecules. That's why nucleic acid are expected to alternative of antibodies. So the unique point is aptamers can be generated by in vitro selection system, repetitive selection and PCR amplification cycles by using nucleic acid libraries with random sequences. And the strong point is relative to antibodies, the aptamer is high specificity and low immunogenicity and easy quality control and modifications for mass production because at first we determine the sequence by the selects. But after that, we can chemically synthesize of the aptamers. So that's why it's very easy to apply to diagnostic and therapeutics applications. But the weak point is the affinity is relatively low as compared to antibodies. This is because only 4 bases compared to 20 amino acids. That is a problem. So another one is low stability against nucleases. I explained first these problems. So this is another science fiction movies. I like science fiction movies, and this is also very interesting because someday simple bacteria came from universe to Earth, and they only few days they evolved to higher creatures. And the scientists determined their genetic materials, and they have 10 bases, 10 different bases. So this is just science fiction, but I imagine this is very interesting because increase the complexity by increasing letters. So if so, rapid evolution is possible, and also increased functionality. So that's why we try to evolutionary engineering method of selection by increasing the DNA letters. in the libraries. So we performed selection procedures using the increased letter libraries. We just add the DS bases into the library as a fifth base. There are two reasons. One is DS base is very hydrophobic. Nucleic acid is generally very hydrophilic, so that it's just a bit difficult to bind to hydrophobic part of the proteins. So that's why we add the hydrophobic DS bases into the library like this. So another one is we didn't add the PX base in the library as a sixth base because DNA is always complementary paired each other. So in this case, DNA has just like a simple to form the single duplex. This is not good for aptamers to increase structural diversity. And if you add the only DS base cannot pair other bases. So that's why maybe the structure might be increased. So these are two reasons. Then we make this DNA library with five different bases and performed selection. And after that, we performed PCR amplification. In this case, we add the DS and PX substrate. And we make a double-stranded DNA. Then we separated only DS-containing bases as next round library. So first, our demonstration, we choose BGF 165. So this is very important for aptamer researchers because only one modified RNA aptamer named MacGen was approved as a treatment for age-related macular degeneration. Only one aptamers are approved. So that is a problem. So that our first target is this. And this is actually, so this is a MacGen. So the KD value is relatively high, 5 to 100 picomoles. And this is RNA. And another DNA aptamer was generated by Larry Gold group. And in this case, it's 300 picomole. And it has two structures, so that another one is much more lower affinities. So then we performed in vitro selection targeting BGF 165. We performed 7 rounds of selection like this. We also add the competitor of the Larry Gold's aptamers like this. And the important thing is we performed more than 150 cycles of PCR in total. So if the natural bases are removed during the PCR amplification, that selection was maybe failed. But most unnatural bases survived during the selection procedures. Then we got DNA aptamers. It has 47 DNA, and only 2 unnatural bases existed here and here. So the KD value is less than 1 picomolar. In the same conditions, the Larry Gold's DNA aptamer is 100 picomole. So that's why only 2 unnatural bases increased by more than 100 times higher affinity relative to the conventional DNA aptamers. Then we replaced the DS base with A because DS base is sometimes often replaced with A during PCR amplification. So that's why we also make A mutant. In this case, the KD value is 300 picomole. So that's why only 2 unnatural bases effectively increase binding affinities. So, and this aptamer also selectively bind to VEGF 165. It cannot bind to VEGF 121 and other proteins. So that's why this aptamer, the selectivity is also very high. So now we have several aptamers by using this method as well. Anti-VEGF aptamer here, and anti-interferon gamma aptamer, and also anti-VEGF aptamers. All the 38 or 75 picomole, which is much higher than the conventional DNA aptamers. And recently, Dr. Howard Young of National Cancer Institute is interested in this interferon gamma aptamers. They have an assay system of the using a culture cells. Interferon gamma interact with the interferon gamma receptor, stimulates STAT1 phosphorylation. And they have an assay system using a flow cytometry like this. So if we add our aptamer into the system, this aptamer can inhibit the interaction so that we tested. 100 nanogram per milliliter of aptamers are completely, so you can see completely inhibit the interferon gamma and the receptor interaction like this. And as a control, we also tested using conventional DNA aptamers already reported. And in this case, more than 100 nanogram of picomole, it cannot inhibit the interferon gamma activity at all. So, but another problem is even if we use this natural basis, the DNA aptamer cannot stable against nucleases. This is a gel electrophoresis of aptamers in the serum. After 1 day, the most DNA aptamers are degraded in the serum. So, but we have another technology using, we call it mini hairpin DNA. This is just natural DNA hairpin structures. If you add the 3' region of this mini hairpin like this, and also, actually we found these two DS bases are very important in this case. This is not so important, so that's why we replaced this region with mini hairpin DNA. So if we modify like this story, the aptamers stability is significantly increased. More than 80% survived after 3 days in the serum at 37 degrees. And also this aptamer can show the in vitro activities. So I will just talk about the mini hairpin DNA structures because we still don't know the simple DNA sequences. So I also try to make a natural base incorporated DNA, artificial DNA, but during our experiment, we still don't know the simple natural DNA. So I just introduce about this for new synthetic biology. So actually, 25 years ago, I accidentally found this unique DNA sequences. So this is a gel electrophoresis of this 21 chemical synthesized fragment. This is a complementary sequence. And complementary sequence shows usual gel mobility on denaturing gels. But this 21 shows always faster mobility by 1 or 2 bases This is 25 years ago, so that at that time, even in chemical synthesis of DNA is also proceeding, so that these are old methods. But when we use several methods, the purified DNA shows this position on the gel. At first, we didn't know what happened to this DNA fragment. So that's why we made a much shorter DNA fragment from the 3' terminus of this 21 mer. So in this case, 1, 2, 3, 4, 5 mer like this, and 6 mer like this, and 7, 8, 9. So if you make this fragment, so this fragment shows higher mobility. After that, increase like this. So that's why the 21 mer shows a lower mobility like this. And we also performed this experiment from the 5' region. Then we determined the GCG AAAGC sequence shows this abnormal mobility. This is thermal stability. This fragment shows TM value is more than 82 degrees. And if you add the 7 molar urea, even in the 7 molar urea, the TM value is 570 degrees. So that's why even in the denatured gel, this fragment is still forms some structures. That's why the mobility is high. After that, we determined the structure by NMR and also GCG NAGC or GCG NAGC shows a very stable structure. Then we call it a mini hairpin structure like this. So this is NMR structures, tertiary structures by determined by NMR, and 2 GC pairs and 1 shared type GA pair. And this is 1A is stacked with neighboring bases. So the important thing is this thermal stable mini hairpin DNA also resist against nucleases. These are very stable against nucleus P1 or nucleus S1. So that's why if we add the 3' terminus of the aptamer, this structure blocked against the 3' exonuclease activity. And also if you add the internal structure of the aptamer, the aptamer imparts higher increased structure stability. So then total structure is very stable even against nucleases. So last part, I will just talk about self-select using our natural base pair selection systems. Aptamer's target are not only proteins, but also cells. So we performed in vitro selection against breast cancer cells. In this case, we just use cells as a target for the selection. After 7th round selection, we can see the gradually concentrates libraries like this. Then we determined we obtained DNA aptamers that specifically bind to MCF7 like this. If we change the DS base to A, so this aptamer's activity is completely lost. So this is gel imaging using these aptamers. We also have another aptamers with the same selection, and so the cells can specifically die with these aptamers. And by confocal imaging, we also know this aptamer was going to cells, maybe lysosome part. So we also see that from the pictures. So that's why this aptamer can be used for diagnostics like PET or something, or it can be also used as a DDS. So finally, I just talk about myself. Actually, I cannot believe I'm in now Singapore. I was 18 hours working in Japan at Riken Institute, but our group now moved to Singapore Institute of Bioengineering Nanotechnology, and in this also like this, and. In this institute, we're focusing on the diagnostics of using these aptamers, and also to expand, to increase the studies of this genetic alphabet expansion systems. And we have also venture companies, Diagnostics Biotechnologies, is still in Japan, and they are focusing on therapeutic applications of these aptamers. So now we are, to expand the genetic alphabet, we are expand our research globally, so that if you are interested in our research, please collaborate with each of ours. So, and this study is also Matsunaga and Michiko are working with here. So, and I also thank Howard Young. Thank you very much.
Thank you, Maria. It was a wonderful initial flavor of everything that xenobiology can do for us. And we'll have various presentations out of the meeting on that topic. Urgent questions.
Natural DNAs are well known for the so-called second Chargaff's or parity Chargaff rule. So what happens with this constraint in your synthetic artificial sequences? Does it break or complete?
Actually, we don't know, but our natural base pair has no hydrogen bonding interactions. So that's why we cannot add a lot of this third pair into DNA. So maybe in this case, it cannot be work. Is it okay? Is it not?
I understand what you say, but it's not okay for me.
Okay.
Force applies for any number of base pairs. So A equals T, in duplex G equals C, and on the DS equals P.
Parity rule actually counts the oligomers over the same strand. No, it knows nothing about the opposite one. Trust me, I pretty
Really?
well know what I'm speaking about.
In the same strand?
Yeah, sure. That complementary palindromes counted over the same strand actually exhibit pretty close frequencies.
Okay. Maybe Fleur Romesberg made bacteria using a third base pair, so they will show the Chargaff rule is correct or
That's
not.
the question. To meet the constraint, you are to introduce not a single couple, but two couples of artificial bases, you see. It's a trick.
Okay.
Okay, thank you so much again to the speaker.
Okay.